I would like to welcome you all to the second webinar in our series of blue carbon in polar ecosystems. The webinar series is organized by two EU projects POMP and SEA-Quester. And my name is Maria Koski, and I'm the scientific coordinator of SEA-Quester. And first, I have a few things I would like to point out. If you have questions to the speaker of the day, then please write them into the questions and answers box. You find it below in the screen. It just says Q&A. And after Christof's talk, I will read up the questions, and and then we can keep track on that. And the other thing I wanted to say that the recording of the talk will be available afterwards in different social media. So then it's only left to introduce the speaker of the day. That is Associate Professor Christof Pearce from the Institute of Geosciences in the Aarhus University. And he's particularly interested in looking into the changes related to how the changes what we are seeing in ecosystems and climate in these days relate to the past changes. He looks at the reconstruction of past climate in the North Atlantic and Arctic, specifically during the last glacial interglacial cycle. And then how does that, in a way, represent the changes what we are seeing today? And Christof did a PhD in Aarhus University at the beginning of times, a couple of years ago now, I guess. And afterwards, he did a PhD, he did a postdoc in Stockholm, and then returned to Aarhus, where he was an assistant professor first, and now associate professor already for several years. And the title, finally, of his presentation is Paleoceanography and Long-Term Carbon Burial in Sediments. So we look very much forward to that. Thank you, Christof, for agreeing on giving this talk, and the screen is yours.
All right, thank you very much. I hope you can hear me well. I'll just find my link to share my screen and choose the right one.
We can definitely hear you, and we can see your screen now.
Excellent. Just give me 2 seconds to get all my windows in the right place. Very good. Yeah, so thank you so much for having me in the seminar series. Thanks, Maria, for introducing, and Paige for organizing, and everyone else who's been part of running this series. It was great to be part of the last speakers, also Dora, who's also here today. A fantastic presentation. Mine is going to be very different. Actually, I'm going to talk about paleoceanography. And I guess the biggest difference here is the time scale. So obviously, if we are going back in time much further than observations and instrumental records, and you can't just win that without losing something as well. So we are also going to lose a lot of resolution. So we are working much further back in time, but then our sampling resolution is a lot lower. And I think therefore also it can be sometimes intimidating a bit to work and to talk to an audience like you, because you are working with satellite observations, with ship-based measurements, real-time observations, long time series for you, or maybe every year going back to the same place, or a seasonal time series. And then you are working very hard with huge teams to try and understand all these processes that we are looking at when we look at blue carbon in the Arctic. And then people like us come along, and we stick a stick in the mud and tell you what it was like 8,000 years ago. So of course there's a lot of limitations also, and I'll be open about those. But I also hope I can convince you about the value of all this work in paleoclimatology and paleoceanography in general. So not only do we suffer from this low resolution, but we also have only indirect measurements, right? There's no thermometers really to go further back in time. We rely on proxy analysis. And I mean, now I'm just chatting only on my cover slide here, but I'll tell you a little bit more about paleoceanography in a second. I'll tell you what time period I'm going to focus on in this research and in this talk and specifically. And then a bit of details and background on what kind of methods we use. So how do we collect these archives, and what kind of tools and proxies can we use to analyze them? And of course, I'll end with a few case studies as well. So yeah, thanks so much, and I hope that you'll enjoy these slides that I'm going to go through in the beginning here. So if I haven't convinced you yet that the paleo is so important, I just wanted to highlight this last edition of the Physical Science Basis from IPCC. So if we have to look at any kind of big authority in climate change reviews of research, the IPCC report here from 2021 in almost 4,000 pages mentions paleo about just under 1,000 times here. So if we are looking at modern climate change and understanding that and then predicting into the future, we really need to have this paleoclimate science for providing a long-term context for observed changes of the past 150 years and projected changes in the 21st century. Then also, paleoclimate reference periods help narrow the uncertainty range and the overall assessment of Earth's sensitivity to climate forcing. And I think, yeah, this is very much motivated by looking at, for example, the temperature response to CO2 increase or doubling. We could just as much interpret this as, for example, efficiency of carbon burial in different environmental scenarios in the Arctic. So the paleoclimate reference period specifically can help us with that. IPCC also mentioned that paleoclimate reconstructions from natural archives have enabled these robust reconstructions of spatial and temporal patterns on multiple timescales. So again, we can also go to different locations at the same time on these long timescales. And then, I mean, a bit sadly, they mention also that many of these paleoclimate archives, including tropical glaciers and modern natural archives like corals and trees, are rapidly disappearing owing to a host of pressures, including increasing temperatures. So basically, if you work with ice cores and you use those to reconstruct paleoclimate, it's actually the warming and the climate change that is then making those archives disappear. That's not so relevant for marine sediments that I'm going to talk about today, but still it's one of the key points that there's some urgency to looking at these paleoclimate records. Now, I'm going to focus on this time period and this time period alone. And there's a good reason for that, and I hope you'll follow that. We can go back millions of years, we can go back hundreds of millions of years, and there's all kinds of interesting time periods of reference that we can use to study. But we are just going to focus on the so-called Holocene, which is the last 12,000 years. And that's the current interglacial period. On the right here, we have a little bit of the geological timescale. You can see that the Holocene is covering, together with the Pleistocene, together they form the Quaternary, so the period of the ice ages, where we have cyclical variations in ice sheets on land and over the oceans, with about 100,000 year cycles or so in the last million years. But we are just looking at the last 12,000 years, which is this short interval that we call an interglacial. So typically it would be between big glaciations in which you have stable climate. That's really key to this period. If we look at glacial climates, there's much more rapid fluctuations, much higher amplitude swings in temperatures and ocean circulation changes. But CO2 has been relatively stable during the Holocene until, of course, recently. And for these 12,000 years, we have relatively calm climate. It's an interesting period to study because everything else is kind of as it is today. So when we look at reference periods in the past, we might have similar CO2 concentrations like we are going into, or like we are already now if we go back millions of years. But then there might be other configurations of ice sheets or even major gateways that have opened or closed. So we use the Holocene also as a period for which kind of the boundary conditions are similar as today. And then most importantly, perhaps, it is, of course, the most recent time period. So no matter where we go, no matter what sediments we get from what lake or from what shallow ice core or shallow marine site, we'll get Holocene sediments or archives that represent the Holocene. So it's really well studied, and there's a lot to say about that. Now, I've said the Holocene is very stable, but it's not boring. If we zoom in, of course, there's quite a bit of change also happening in this Holocene climate. There's a lot of studies that try to make these kind of global mean temperature composites. Here's just one of the more recent ones where we see that during these last, say, 10,000 years or so, we have this relatively stable climate. But of course, there's some variability. Strikingly, of course, is the most recent change that you see all the way in the right of this graph. I guess you don't really see my mouse. maybe if I change the laser pointer here, yeah. So over here you see this unprecedented, both in magnitude and in rate of change, the recent increase in temperature. But we also see, if we go further back into the early and mid-Holocene, that we have this period of elevated temperatures, depending on exactly which model that we're looking at. Now, this is more extreme when we go to higher latitudes. So on the left, we have global temperatures, and on the right, we have that divided into 3 different latitudinal bands. So if we're in the Arctic, we see these changes quite a lot clearer than we do as we are closer to the equator and the panel in the bottom right. Just like today, where Arctic amplification is responsible for maybe a threefold amplitude of the signal of global warming that we see on average, we see that also when we go back in time. Now, we've talked about paleoclimatology, the Holocene, some introduction. Let's turn to the archives of choice. And of course, I already told you this, when we look at blue carbon, we should look at seafloor sediments. So we're not going to talk much about ice cores or anything like that. But when we do paleoceanography and paleoclimatology, we work with these seafloor sediments. And in this schematic of the marine cryosphere, that could be any kind of environment offshore Greenland, where we have different components of the cryosphere on land, so these ice sheets ending in big ice shelves, so ice streams producing lots of icebergs. And then also, of course, the sea ice in these fjords and close to the shore. All of these leave their imprints on these seafloor sediments. And as they accumulate layer by layer, we can go further back in time and reconstruct all these kind of physical components of the marine environment and the cryosphere. So it is really low tech. We come in with our research vessels and simply punch a hole through the mud and pull it back up on board and study those sedimentary layers. And I guess you've seen all these. I just wanted to take a minute and walk through them very quickly. But on the left, we have our box score that many of you are using also when you're looking with benthic processes and used a lot for biology studies as well. We look a little bit more into geology when we start to look at this rumor core that goes further back in time or further back in depth of the sediment, but it keeps a pristine surface, so we can use that for looking at modern conditions and then going back a few hundred, maybe a few thousand years, and depending on the sedimentation rates. And then if we want to go further back, and especially if we want to study the Holocene in this example, then we use our so-called gravity core. So that's just an extension of this rumor core, really low tech, about a ton of weight, a ton of lead of weight on top, and then a 6-meter steel barrel that can be extended to 10 meters or so. But we don't need more than that to be able to core through these roughly 10,000 years back in time on the shelf around Greenland. So low tech, no drilling required, no fancy release mechanisms or piston coring systems. And that's another reason why it's relatively easy to go and access this time period of the Holocene. A few more pictures to illustrate some of these examples. On the left here, we have a beautiful rumor core that's just been brought on board, where you see this undisturbed fluffy surface on top. So that's showing us the sediments that have just been deposited that correspond to basically the day or the month or the year or the season in which the sample was taken, and then going back in time. And on the right, some of these sections of these longer gravity cores that we have to handle and split up into smaller sections to be able to store and analyze. And we've been doing that for a while around the Greenland and in the North Atlantic. As you can see, all those orange dots on the maps, they are the cores that we have in our repository here in Aarhus. And many of these have been cored thanks to these cruises that were part of these interdisciplinary projects like SEA-Quester and like Ecotip also before. Just some more pretty pictures of sediment cores to show you a little bit what they look like. But then you can also see that the sediments here, as we look at them here on this freshly cut surface on the left, they are pretty boring, right? There's not much to see from those sediments. We really did rely on a lot of different analysis We need to take those grains and look at them by shooting a laser at them to get the grain size distribution, or extract them for biomarkers, or sieve them for microfossils to be able to tell something about the past environment. And I'll get to these proxies in a little bit more detail in a second, but basically what we are doing, and also what we are doing in part of all these other projects, POMP and SEA-Quester, is really trying to figure out all these different components of the carbon export and carbon burial and sequestration into the deep ocean and eventually into the sediments. And whilst many of you are working on figuring out the size of all these arrows and movement from carbon from one pool to the other, and the behavior of all these components of these ecosystems, I will focus here a bit on a slightly simpler approach and just look at what's in the sediments. And then what's in the sediments, and that's my idealized simple graph here on the right here, kind of making a shortcut from all these interactions and arrows on the left. And my point is here really that marine sediments and the tools that we are using are integrating all of these processes over a longer time scale. So when we measure organic carbon in sediments and we assign this ages, we can look at burial rates of carbon, obviously on long time scales, and we've shortcutted all these processes of remineralization or fluxes upwards. We are just looking at what's buried below the reactive sediments in the upper layers. Of course, what we then need to figure out is how much of this belongs to this so-called blue arrow here on the right, this atmospheric carbon and primary productivity that is buried in these sediments compared to actual reworked terrigenous organic carbon, because not all that carbon that's buried in the sediments obviously is coming directly from the atmosphere as true sequestration. And that's really what we are trying to work on in combination with all these different proxies on sediment cores. And so I'll highlight a few of these proxies and I'll talk to you about some of them in a little bit more detail. But in general, as we take these cores up, we look at the very basics, these non-destructive analysis that include just looking at the color or the lithology, the description of these cores, the physical properties, the density variability, the magnetic susceptibility, CT scans, radiography, looking at what's inside the core. Looking at basic stuff like the grain size distribution can already tell us a lot about the bottom water currents, for example, so the energy that's in the water, but also the abundance of icebergs, for example, that distribute these ice rafted debris particles into our sediments. We do a lot of geochemistry. Still, when we're looking at non-destructive scanning, we actually do XRF elemental composition scans of these sediment cores, so we can look at the variability of different elements in the sediments. And obviously, also, as geoscientists, we look at the mineralogy to some extent to look at the provenance of the grains that are ending up in the sediments. I'll talk a little bit more about some of these biomarkers, but we use different tools to reconstruct temperatures in the past or sea ice cover. Organic carbon, I've already mentioned, and I'll talk much more about that in a minute. Elemental ratios that can tell us something about temperatures, for example, and then all these isotopes that are recorded in the microfossils themselves or in the sediments in general. And pyrolysis I'll also touch on a little bit more in a second. Micropaleontology, I'll just expand on a little bit because we use that really a lot, and it's one of the key proxies that we study in paleoceanography traditionally, but also in this department that's been going on for about 50 years or so, the work on this. And there's a constant renewal of looking at maybe more DNA approaches or more biomarkers, but we keep coming back also to these actual fossils. And you'll see here on the graph on the right is that our view of these ecosystems and the different microorganisms that we are studying is very much biased towards those that actually produce fossils, right? Because that's what we see in the geological record. So we are very well aware of maybe that only even in diatoms or in forams, just a few percentage of what's actually in the water column make it into the sediments. but still that's the view that we have on the past, to look at the assemblages of these both planktic and benthic microfossils. They can be used to tell us about salinity, water temperatures, ocean currents and water masses and so forth, sea ice cover, and of course also primary productivity to some degree. And at the same time, they also record for us the chemistry of the water. This can be in the form of isotopes or trace metals and ratios of trace metals that we analyze these specifically forams on very much. So we rely very much on these microfossils. And as I mentioned, also on biomarkers, and there's just one I'll talk to you about a little bit more here. That's this IP25, which is the catchy name, ice proxy with 25 carbon atoms. So it's an organic compound that is biosynthesized by sea ice algae, by diatoms more specifically. And so what's interesting about this specific biomarker is that many of the diatoms that produce these biomarkers or that live in the sea ice don't actually make it all the way into the sediments. So they would dissolve, or they would be already disappearing before they get buried in the sediments, whilst this marker, this highly branched isoprenoid, actually makes it into the marine sediments. So we can use that and that presence for telling us that there was seasonal sea ice cover in the past. Yeah, I can talk about these proxies all day, but I guess if I summarize a little bit, what we're trying to reconstruct here with all these different tools is we could be in any of these scenarios. If we look at the physical environment, again, if I go back to this variability of ice sheets, extent of floating ice shelves, iceberg activity, sea ice, all of that is recorded in these underlying sediments, either by things that we can measure in grain size, like ice rafted debris, or these subglacial plumes that we can figure out from mineralogy or from elemental composition. And then, of course, we can look at the microfossils to look at the ecosystem. And what we've really learned from working together in groups like this, again, is that we have to be more careful when we try to reconstruct the physical environment and climate, and then the ecosystems and carbon burial, and keep those things separated. So in many ways, we've maybe used the micropaleontology, so the remains of life, basically to reconstruct the physical environment, but then we can't really say much about the ecosystem because we go in this kind of a circular loop of reconstructing. So we're trying more and more to keep these worlds separated, so we can use maybe the lithology, CT scans, grain size to talk about where we are in the past in terms of ice and the physical environment, and then look at the carbon and the microfossils to look at the ecosystems. And of course, I should mention bioturbation as well, because I mean that is also a really important factor, not only for carbon burial and for many of the processes that you all are measuring, but for us, it's just the most annoying thing, right? It mixes up all these sedimentary layers. It ruins our stratigraphy. It mixes the signal from one year to the other, or in low sedimentation rates, it might even mix it over hundreds of years. And we're really trying to now also get a grip on that and figure out how we can use it to our benefit by looking at CT scans of sediment cores and reconstructing burrows and bioturbation activity related to the environment. It's a long intro to get to a few examples here, and I'll walk you through a few case studies of Holocene organic carbon in sediments that we've been studying in recent years in our group. And this first one is from East Greenland and from a core called 171G. That doesn't matter so much, but many of you in these projects will recognize exactly what place this is. This is just outside of Young Sound. So that's on the East Greenland shelf, right in front of that sound that's been studied so much in these projects, and especially at Aarhus University. In this place here in 171G, we've retrieved a sediment core that you can see here on the left. That's about just over 4 meters long. There's quite a bit of variability in the image, but not that much. But when we start to do radiocarbon dating on these, we can start to look at the so-called age depth model, or the chronology of these sediment cores. So how far back in time do these sediments go? Well, I promised you to look at the Holocene, and that's what we're showing you also here. So you can see on this on the x-axis, sorry, in the bottom, the modeled age. So that's 12,000 years before present, or kilo years. And on the y-axis here, we have depth in the record. So we're going back just to the onset of the Holocene, approximately here. There's some uncertainty, of course, associated with that. And we have very high sediment accumulation rates up until about 30 centimeters below the surface, when we are only about 8,000 years before present. That's when the sedimentation rate rapidly declines, and we have basically the rest of those 8,000 years just in that upper 25, 30 centimeters of sediments. So we can see already there's a huge variation in sedimentation rate in these sites. That's going to have big implications for the burial as well. And if we start to look at some of these carbon analysis that our colleagues here in the department are doing in the form of pyrolysis analysis here, we can see how much of this carbon is actually labile and how much of this is inert organic carbon. And with that, meaning that it is actually reworked or older organic carbon that's not directly available and is not coming from fresh production or from primary productivity. In this specific case, we can see that the TOC that's measured in this core over the entire Holocene, it doesn't fluctuate that much. Actually, the fluctuations are more of a case of our instrumental precision, but generally we have about 1% of all these sediments that are organic carbon. And that's about also where it fluctuates around, so 1%. But then when we do the characterization of that carbon, it turns out that 90% of that is inert or reworked organic carbon. So in this very specific case, the site we can conclude is mainly a sink of recycled carbon from land, and all this TOC that's buried in the sediments is not equaling new drawdown or actual sequestration. It's only a very, very small component of that. This is done with pyrolysis. We can also do this a little bit more in the classic micropaleontological ways. In this case, in that same study, looking at dinoflagellate cysts, actually. And this is outside of my expertise, really, but I understand the idea of using these old versus new ones. And that is that although many of the dinoflagellate cysts that are found in the sediment cores are belonging to recent assemblages, that means that they were coming from primary productivity, that there's also quite a significant component of them from Cretaceous outcrops. So Cretaceous meaning 100 million years old sediments that are outcropping somewhere else. And we can again see that there's quite a good component of that, and that's variable through time, of old organic matter coming into these systems. Now, this is not as complete as the previous graph I just showed you, because this is just showing you one group of microorganisms. But again, it shows us that there's a lot of reworked material, and it's a way of applying our more classical techniques to this idea of quantifying inputs of reworked organic carbon. If we go to the other side of Greenland for another case here on northwest Greenland, this is a study where we used radiocarbon offsets as an indicator of the age of carbon. That might be completely illogical to you. For us, the radiocarbon is something that we use to find out how old these sediments are, right? So we would normally use this analyzed preferably on foraminifera or other organisms that are incorporating the age or the 14C age of the surface waters, because that would give us the best chronology down core. But in this case study from a core AMD 204C from just offshore Upernavik, I'll show you the chronology of that core in this slide here. The blue line here now going down core, and just on the bottom here, we have now the age again in years before present. So we are going back again almost to 12,000, so almost the entire Holocene. The blue line here connects these ages on foraminifera. So those are measurements of the age of the forams in the core, whilst the green observations here, those are radiocarbon measurements on the bulk organic carbon in the sediments. So there's a very clear offset, and the bulk organic carbon in the sediments is constant. Oh, it's not constant, but it's always older than the true age of these sediments. And this is very interesting because we can see that there's about a 3,000-year age difference between the bulk organic carbon and these foraminifera. So a lot of the carbon that's ending up in these sediment cores is also something that's much older than the primary productivity that we're trying to measure here. We can use, in this case, also the variability of this offset to look at the flux of reworked old carbon into these systems. And we can see through time in the panel B here that if we plot the age offset between the carbon, the bulk organic carbon, and the forams, from 4,000 years up to maybe 1,000, 2,000 years in the very top of the core. So we have periods during which we have much more input from land, also when glaciers have been expanded and we have much more effective kind of erosion from land, where we have a higher offset. Finally, another case study from Southwest Greenland, where colleagues worked on a core that we took from offshore Godopsfjord or the Nuuk Fjord system here in southwestern Greenland, where this combination of radiocarbon and 13C was used to characterize the different pools or the different end members of the organic carbon in the sediments, and then used to reconstruct that through time, again through the entire Holocene. And we can see here that there's quite a bit of variability, again, just like in the previous study, where we have higher and lower shares of the input of marine organic carbon versus terrestrial organic carbon throughout different phases of the position of the ice margin in the past. And with that final case study here, I'll go into some conclusions. And I want to say that so these marine sediment records that we've been studying, they are providing a good record of carbon burial. But of course, provenance studies are crucial. If we can measure all the carbon we like in these sediment cores and we don't know exactly where it's coming from, there's not much to be said about sequestration or burial. We do pyrolysis analysis. We look at reworked microfossils. We look at the radiocarbon ages of the bulk organic carbon. And we use biomarkers for looking at these differences as well. And then very importantly, these marine sediments, like I was showing you in the beginning, they kind of integrate all those different arrows in the complex biological carbon pump and cover everything from productivity, preservation, and remineralization, and of course the long time as well that is all covered into these sedimentary samples. The signal is heterogeneous and can be highly local. That's unfortunately the case. If we go to different places on the shelf, we get different signals, and that also emphasizes that we cannot make these huge inferments, or how do we say, we cannot scale it up so well if we don't have more sediment cores, right? So we know that depending on the depositional environment, we will have much different carbon burial stories, even at regional scales. The Holocene variability of this organic carbon burial is, of course, driven by climate and things like ice extent, but the overall variability is relatively minimal. It's maybe smaller than we'd expect. And not surprisingly, sediment accumulation rates are one of the primary drivers on the carbon burial fluxes. So we've seen that especially in the other core, where we have a relatively stable amount of organic carbon throughout the whole 12,000 years, but that's in percentage of sediments. And if we convert that to fluxes, it will be heavily influenced by the accumulation rate, of course. And with that, I'll finish up here and say thank you so much for listening, and looking forward to some discussions and questions.
Thank you, Christof. That was really great. There are at the moment no open questions, so please write your questions into the questions and answers box. And meanwhile, actually that gives me a chance of asking a few things of my own, just to, keep it going until people type it in. So the first thing I wanted to ask you is that, what do you know what influences this old versus new carbon ratio? Can you say something about that? When would we expect a lot of actual sequestration to happen?
Yeah, so in periods of higher productivity, we would have more sequestration going on. But then also we have the ice margins that play a big role if we have a lot of influence of reworked material from land by erosion that is simply more effective when we have ice streams over land. So those components would be very important for driving those differences. Yeah.
And can you see that in the Holocene records, this sort of changes?
Yes. So we can see that when there is an ice margin nearby, and we have huge input of terrestrial material, for example, that means we also would have much higher sediment accumulation rates, and meaning that we would have more rapid burial. So we would have more input from land, but we'd actually also have more sequestration or more burial from primary productivity in these times.
All right, thank you. Yes, now we have a question from Paul Renaud. He is writing, maybe I missed it in the figures, but did you calculate carbon accumulation rates in the course and perhaps linking changes in this with any evidence of changes in ice cover, primary production, or other processes?
Yes, but I don't have those graphs or those slides at the moment here, so sorry about that. I think it's one of the key things that we are going to deliver in SEA-Quester is to make these integrated maps of all the cores that have been analyzed around Greenland and exactly produce those fluxes, so the carbon accumulation rates through time. So it is primarily driven again by the sediment accumulation rates, because the organic carbon content of many of these sediment cores doesn't fluctuate that much as we see in the burial rates.
Okay, then we have a question from Andy Fisher. Is there any way to use O2, oxygen, as a proxy, as a means of disaggregating old and new organic matter?
I don't know. It could be worth looking into. I think maybe with biomarkers we could look at you mean oxidized molecules or ratios between fresh and old ones, but I don't know how to use. We could maybe look at the benthic ecosystems, of course, to reconstruct oxygen in the bottom waters or in the sediments, but that would again add one more layer of uncertainty on it. But yeah, that would be a way to test. But I'd love to hear ideas on that.
So that's something to look into.
Yeah, I'm writing that down.
Yeah, actually, okay, Andy is pointing out that you will talk.
Very good, Andy.
I'll hope so.
We definitely need to.
So while we wait, if someone still has a question while these are typed in, I have another question actually, a little bit about what you always wanted to know about paleoceanography but didn't dare to ask. You because you mentioned that you don't really use anymore the organisms as or you try to separate these two things, the environment and the organism. But I have always been wondering when you use forams or radiolarians or diatoms as an indicator of environmental change, haven't these organisms been adapting or changing to their environment over the years? So when if we know a foraminiferan X now prefers these environmental conditions, how do we know that they did the same 10,000 years ago?
Yeah, that's a great question. I guess that's one of the central assumptions that we use in paleoceanography. And I think that on the scale of the Holocene, again, that we are much safer to say those things than going back millions of years. I mean, our colleagues who are working with plant fossils of plants that are simply not there anymore, right, or animals, then we have a lot more of those problems. But we work with assemblages, so counting the different species and using modern analogs, and that's the best we can do for those cases. So, I mean, it's a good point. We assume that the species that like it salt and warm, or vice versa today, are the same ones as 10,000 years ago.
Okay, thank you. So now the final possibility to ask questions, and if nothing is coming within the next 10 seconds, then I think we will thank.
I think I wasn't sure if Marit Solveig had a question earlier, so if you wanted to speak up, Marit Solveig, you can do so now.
I had no question. No.
Okay, sorry. I had a quick question.
Yeah, sure.
I was wondering, I've studied sediment cores in a freshwater context before, and their pollen grains were also really important, but I don't know if that's true in the marine context. Is that something else that you can pull out? Yeah.
There's not much in the marine sediments that we are looking at. They would also require a different way of preparing them than what we normally do. But yeah, in dinoflagellates preparations, they would show up. I think we are not doing that much kind of terrestrial vegetation reconstructions, but in these marine systems, they can be used for long distance atmospheric transport pathways also. So, but then it would be really, quite uncertain about exactly what area we're reconstructing the vegetation from.
Okay, thanks. Maria, you can have the floor back. Thanks.
All right. Thank you. There is no open questions and no hands up. So I think then we just say big thanks for Christof for this fantastic presentation. And thank you for all of you who attended. The recording will be made available later. And we hope to meet as many as you as possible in the next seminar, which is in 1 month.